go.
I go. Great. Hello, class. This is my team.
I'm Timmy.
I'm Luis.
And I'm Michael Ontiveros. And we are presenting the topic of biochips for our advanced topics technology presentation.
All right. So let's go.
Okay. So the definition of a biochip. They're also known as microarrays. It is intended to function in a biological environment, especially inside a living organism. So this is just the pure and clear textbook definition of a biochip.
So the history of biochips. After the discovery of sensor technology, Hughes in the year 1922 created an impact by inventing the chemistry-based sensor with the help of a glass pH electrode. Now, this sensor technology was incorporated in the DNA-based genetic researches to attain a complete analytical study of the DNA components. This was when biochip came to existence as a new technology. Then Karen Mullis in the year 1983 invented a component in living organisms which are considered as their official carrier of genetic information. This component was termed as DNA, which was visible in the polymerase chain reaction technique first conducted by him. And later in 1986, Hood and his co-scientists, in order to detect hybridization of DNA molecules optically, invented a process labeled DNA molecules with fluorescent marking instead of radio marking. Next slide. Okay. Now, the history from today. It is important to realize that a biochip is not a single product, but rather a family of products that form a technology platform. Although the many developments over the past 2 or 3 decades have contributed to its evolution. Though the first biochip was invented by an American company, namely called Affymetrix. They were the ones who essentially paved the way for many of the new biochips coming out with their biochip called gene chip. So DNA chips were the first biochips and are still dominating the biochip market. And a few years back, Affymetrix led the way with the gene chip. Like I said, they often had to enlist support of colleagues in other departments to analyze, view, and interpret the data provided by DNA microarrays. With the help of many colleagues and government funding, it can now contain around close to 400,000 sensors, which is a lot easier. And next slide. Then recent developments. So with the drug discovery, microarray technology found in biochips are extensive application in pharmacogenomics. Pharmacogenomics is a study of correlations between therapeutic response to drugs and the genetic profiles of the patients. Comparative analysis of the genes from a disease in a normal cell will help the identification of biochemical constitution of the proteins and synthesized by the disease genes. The researchers can use information of synthesis drugs which combat with disease proteins and reduce their effect. For example, using hepatitis C virus to proliferate one of its proteins needed to bind RNA in a specific way. Using a specific biochip to screen 1,200 small molecules to see if any of them inhibited the protein RNA interaction. Using this method, researchers found 14, and the findings were used to develop drugs that then went to clinical trials. The chip could employ this type of strategy to discover potential drugs to treat many more diseases, including cancer. Labs are already using the chip to grow proteins into the crystalline arrangement necessary for the studying protein structure. The structure of both the Ebola virus and the H5N1 influenza virus were solved using this type of chip. Next slide.
All right, so you got a couple of components when speaking about a biochip. So to start off, you have a transponder, and there's two different types of transponders. There's active and there's passive. An active transponder is when the operator is activated by using a low electric charge, and a passive transponder doesn't contain any of its own energy or battery, so it's technically passive. So basically, with the passive transponder, the doctor or the professional who is is basically scanning you at the time, and then will be able to turn on the biochip from outside. So to get started here, you got microchip, which stores a unique identification UID number that ranges between 10 to 15. You have the antenna coil, which is used to send and receive the signals from the scanner or reader. You got the turning capacitor, which is where the transponder sends the low electric charge to turn on the device, and it is one thousandth of a watt, which it will send to the operator. You got the glass capsule, which basically it just holds everything together. It holds the antenna core, the capacitor, and the microchip itself, and it's made with biocompatible material, namely soda lime glass. And then when you look at the transponder, you also have to look at the reader, and the reader, all it does is carries and receives the ID number or transmitted code sent back from the implanted biochip. So it basically just reads in the information. All right, and now we're going to go ahead and discuss the types of biochips. So there's three different types of biochips. There's protein microarray, DNA biochip, and microfluidic chip. So to begin, let's talk about the protein microarray. So the protein microarray is probably one of the most difficult chips to get right, because you need to go ahead and get a healthy amount of proteins from an individual. And basically what the protein microarray does is to try to treat pre-existing conditions with antibodies and try to go ahead and examine them inside of the body and figure out the best course of action for the disease itself. So a protein microarray or a protein chip method is used to follow both the actions and connections of a protein. The primary goal is to find out their function on a large scale. The main advantage of a protein microarray is that we are able to track a large number of proteins in parallel. And there's five major applications when you're speaking about it. There's diagnostics, proteomics, protein function analysis, antibody characterization, and treatment development. So in diagnostics, what it's doing is it involves detection of antigens and antibodies in the blood sample that you give, and it's a good way to basically profile the disease, new or old, to get a little map of all the proteins inside of the disease itself. Two is proteomics, which basically is a way to express the profiling, which it basically shows us which proteins are expressed in the cell of the disease itself. Three is the protein function analysis, which interacts is basically interacting in a protein way, trying to figure out which kind of what protein needs to be matched with what in order to help it, or in order to create a perfect or better antibodies for the fourth step, which is antibody characterization. And there it specifies and it basically mapping epitopes so it can create antibodies. And then the fifth step is treatment and development. So treatment development, they basically try to figure out the best course of action in order to develop the antibodies for the body itself. And then there's reverse phase protein microarray, also called RPPA, and all that it basically involves complex samples such as tissue lysates. And what happens is that the cells are isolated from various tissues of interest and are laced. The lysate is arrayed onto the microarray and probed with antibodies against the target protein or of interest. So basically, they're using the antibodies they got from the tissues and trying to see which one is the best to fight that specific issue in the body there. And then also there's functional protein microarrays, also called target protein arrays. And basically these protein chips are used to study biochemical activities of the entire proteome in a single experiment. And with DNA microchips, it's actually much more simpler than protein ones because what's going on is more of a test, and it's a test for to see whether you have the disease or not, whether it can be, to go in for, let's say you want to go in for a doctor checkup. Maybe they can do this at the checkup to see if you have any cells in your body that could possibly turn cancerous or become abnormal cells. And what they do is that there's like a set of tiny DNA spots fixed on the surface of the chip itself. And what the doctor is going to do is he's basically going to go ahead and take a blood sample of RNA, and then he's going to go ahead and test it. And then, as you can see, the hair, they have various amount of colors. So the red color means that it indicates that the gene is strongly expressed, and it could become an abnormal gene, which can lead up to cancerous cells. Green means that it indicates that the gene was strongly repressed, meaning that there's less likely for it to have cancerous cells or to turn into an abnormal cell. Yellow spot gene is neither expressed or repressed, so it's kind of they're not sure currently. And then the black spot indicates that the gene is inactive. That gene in your body is basically inactive, it's not turned on. So it's a little bit simpler than protein. It's just really a good way to check and to see where you are at, and also to help those those conditions and see exactly what you need to do in order to stray away from that path. And then you have the most used currently of them all, microfluidic chip. And what it does, it is inserted in someone's body, and then there's a couple of things that's inside of the chip here. You have mostly being a little bit of liquid in there that is like it's basically a very small amount of liquid in there. And then that liquid has things such as, and that liquid is actually used to trap and collect the cancerous cells from the normal blood cells in the body. So basically, it's used to entrap those cells out of there and try to flush them out of your body. So this is good so that it can limit the amount of procedures someone uses, because you know how they have procedures to try to remove the cancerous clots inside of someone's body, where you can actually go and fight the fight from within the cells of the cancer and try to remove them from the body. And this is probably the most one to get implemented currently, because it's at its testing phase right now, and it's really trying to go ahead and see whether it's really trying to take the fight at the cells itself.
Okay, so how are biochips used today? Some applications for biochips include personalized medicines, molecular diagnostics processes, such as like they collect biological markers on an individual's genetic code to perform medical tests for the molecular diagnostics processes. And branching off of that, they could perform medical tests such as drug responses, detection of diseases, and many more. Biochips they can also be used to generally identify a range of biomolecules in any given sample. So we've seen DNA being analyzed, biochips. They can also analyze antibodies to try to find cures to viruses or other diseases, and enzymes to analyze proteins, catalyst protein speeds of breaking down chemical reactions, and even more biomolecules listed, or it can even perform even more actions to other biomolecules. Next slide, please. Okay, so where can they be found? Biochips can be found in many fields, primarily in the health industry. So they're found in biochemistry, and they're used in biochemistry to analyze and record biochemical reactions, mostly for experimental purposes. They're found in microbiology, pharmacogenomics as well. They're used in pharmacogenomics to rapidly perform drug tests to help speed the development and effectiveness of new drugs to treat diseases. And much more related fields as well in the overarching health industry. Next slide. Okay, so the market competition. Biochips are separated into four major regions throughout the world, and they happen to be the largest in the Northern American region. So the four regions are North America, Europe, the Asia Pacific, and the rest of the world. So in the North American regions, it's typically dominated by the United States, and some of the companies involved are Agilent Technologies, GE Healthcare, and Biorad Laboratories, to name a few. It's projected that North America will account for the largest market share, followed by Europe between these regions. So North America, specifically the United States, is expected to lead in the biochips market share. Next slide. So here we have a chart from one of the articles that we researched, and it shows, you can see here, North America, represented in the blue column at the very bottom, is growing and is expected to take up about a little more than a third than the world market of biochips compared to Europe, Asia, and the rest of the world over time. Next slide.
Now we're speaking about the advantages of biochips here. So there's a couple of advantages that we listed, and then mostly in the advantages of it kind of ends the process of time consuming inside of the medical industry. So it can perform thousands of biological reactions in seconds. It's great for those with pre-existing conditions. So it's very small, powerful, and honestly faster than what we are currently doing today. It can simulate change in the health industry, meaning that it can possibly reduce the amount of surgeries people have to take, and also prevent people from getting diseases because we're trying to take the fight to the disease instead of just trying to fight off when it's already brought, right? So and also it's just easier to get to the root of the problem in that individual. You know how every individual person is different. Well, we have to go ahead and try to find the root of the problem in this individual, and then the biochip can help find exactly what protein is off, what DNA particle is different than it should be, and try to insert the correct one into the individual, made to fit an individual and his or her needs. So we all know that doctors definitely work hard, and then they see a lot of patients in a day. Well, each individual person would get a biochip to themselves, meaning that biochip would be just determined to go ahead and just look after them and their needs. So basically be his and hers only. So that's a very strong strength in that there's no really, it's not trying to do multiple things at once. It's just trying to fit your preferences.
Okay, so to elaborate a little more about the advantages, one of the benefits of biochips is that they can perform faster experimentation. So traditional DNA experiments are pretty slow. DNA experiments require a lot of manual intervention without the biochip. So there's a lot of processes and methods that need to be done before you can expect to see any results. However, biochips have helped increase the processing and the speed of experimentation. They're able to perform thousands of reactions in seconds, and as the biochips processing speeds are looking to increase over time, and this speed increases due to the technology becoming more advanced as we develop even faster processors and even smaller processors to develop higher effective and efficient chips. Next slide, please.
And now we're going to go ahead and speak of the weaknesses in biochips. So it raises problems with individual privacy. No current standard of production. Currently, there's many companies trying to create biochips, and right now there's no current standard of what it should include and what it should not include. So right now, until we get that current standard, then that is a weakness currently. Chip could be possibly damaged inside of the the owner's body, and that is also a possibility depending on what the owner is doing. Maybe they might have a couple of restrictions of what you can do with the chip inside of you. We won't know until it's actually implemented. Chips are expensive because they are not currently practical. So right now they're obviously in the testing phases. So you probably couldn't even buy yourself a chip if you could. But I know that as soon as they launch, they're gonna be pretty expensive until they're more, what's the word here? They're being produced more, and more companies start trying to create their own biochip, meaning that it will create competition, which will make the chip cheaper than it originally began with.
So elaborating on one of the weaknesses, one of the greater weaknesses people see is biochips on privacy. People are concerned with their loss of the personal information, as now the biochip can now record your information and send it to a professional without having to personally interact with said professional. Also, it could potentially be used to trace wherever you go, which raises more concerns about privacy and and people afraid that their information may be used without their consent. And so they fear also that it could be used beyond healthcare needs. So this is one of the bigger weaknesses that biochips present amongst its own market. Next slide, please.
All right, expensiveness. Essentially the primary manufacturing challenges is the process of placing each sensor at a specific position, typically on a Cartesian grid, on the substrates. Various means exist to attain this placement, but typically robotic micropipetting or microprinting systems are used to place tiny spots or sensor material on the chip surface. The low throughput nature of this process results in high manufacturing costs. The issue would be for payers in accessing this technology, because microarrays can range from 300 to 1,000. The scanner and supporting equipment cost from 100,000 to 250,000. About half the utilization of microarrays technology is used for cancer research, typically from 3 to 7 microarrays per experiment. Like I said, different uses for biochips can become very expensive. Like in this instance, different biochip price per test with the Hydra 1K biochip, another company's biochip, could be between $7 and $15. That's very inexpensive, but other tests to detect patterns can easily cost, with the most expensive being DNA sequencing, which could cost up to $700,000. And DNA biochips are the popular biochips used in healthcare professionals. Next slide. Then with the breakthroughs in biochip, it could be 3D printed biochips, a different way, a new way of creating them, and as well as next generation sequencing. So with the 3D printed biochips is essentially a new nanoscale printer that allows researchers to imprint more complexity on the surface of biochips than any of the currently available commercial technologies. The method involves exposing a biochip surface to specific organic regions, and then using tightly focused beams of light to the immobilized regions to the chip surface. The process allows scientists to repeatedly expose a single chip to the same different factors and imprint the reactions onto different sections of the biochip. The result is a biochip that can accommodate more probes than it is achievable with current commercial platforms. In addition, benefit of the new tool is that it allows researchers to reliably print on a variety of different delicate materials, including glasses, metals, and lipids, on the length of scale of biological interactions and without the use of a clean room. It also allows scientists to fit more reactive probes into a single chip. These improvements could, in theory, reduce the cost of biochip facilitated research. Then another group of researchers from Seoul National University explored the role of advancements in biochip technology are playing in driving a more groundbreaking scientific discovery through medicine, through the next generation sequencing, meaning a high throughput sequencing. When these biochips meet next generation sequencing, the net value of both technologies will increase exponentially. If biochips that allow single cell analysis prevailed for the last decade, there are many more biochips with different functions that will allow innovation in the biofield, including drug screening biochips. The researchers provided next slide.
We're gonna go ahead and take a quick look. Video here, very quickly.
It is time now for our latest B-West series, Bioengineering the Senses, a closer look at the technology on the frontiers of medical science. Today we take a look at organs on a chip. Will this device speed up and improve the way new medicines are developed? Take a look.
More and more investment has been going into pharmaceutical development and basic scientific research, but less and less drugs have been getting approved.
Could this be the breakthrough that finally flips that equation? Researchers at Harvard University's Wyss Institute call it an organ on a chip. The device, no bigger than a USB stick, has distilled down to its key features what makes an organ an organ. They started by creating a lung, using a method inspired by computer microchip manufacturing, which works at the same size scale as the living cells in our body. The scientists lay down lung cells on a membrane with blood cells moving on the other side. Then they add the magic ingredient. The device is stretchy.
And when you have the suction applied, it stretches the middle channel, and when you release it, it comes back. So it actually breathes back and forth.
The organ on a chip has already won the Design of the Year award from the Design Museum in London, and it's on display at the MoMA in New York. Industry is also taking it for a test drive. Emulate, a startup that was spun out of the beast to commercialize the chips, raised $12 million in Series A funding last year and has partnered with the Janssen subsidiary of Johnson & Johnson to create a liver and a thrombosis on a chip.
Blood clot formation, thrombosis, is a common side effect in several drug classes, including oncology drugs and immunotherapeutics. That can be a very dangerous side effect. The collaboration involves the development of a thrombosis on a chip so we can test the safety of the potential drug candidates.
R&D spending at the 6 biggest pharmaceutical companies exceeded $40 billion in 2014. By comparison, these chips are easy to mass produce, cheaper than testing on animals, and because they're transparent, you can watch what the cells are doing in real time. DARPA, the research arm of the Pentagon, has invested in the chips and is interested to see if 10 organs can be hooked up to each other, creating a human body on a chip, connecting 4 organs so far and striving for 7 by year end. They also have FDA backing to use the chips for testing anti-radiation drugs, something human volunteers can't be used for, and where animals are a poor substitute. And maybe we'll soon be able to put our own cells into these devices as personalized medicine takes off.
All right, so with that, we got a little into what the chips can do. So now we're going to go ahead and continue with this.
So biochips in the health industry, the NA, they're used to monitor patients, do disease diagnostics, as well as predict patients that can become sick as you continuously monitor them. So DNA microarray technology helps researchers learn more about different diseases such as heart disease, mental illness, infectious disease, and especially the study of cancer. Until recently, different types of cancer have been classified on the basis of the organs in which tumors develop. Now, with the evolution of microarray technology, it will be possible for researchers to further classify the types of cancers on the basis of the patterns of gene activities in the tumor cells. This will tremendously help the pharmaceutical community to develop more effective drugs as the treatment strategy will be targeted directly to a specific type of cancer. A biochip can pick up on the body's response as well to different medications. So they include drug toxicity. That is an important function since many patients are taking potentially dangerous medications. By detecting the side effects and altering the dosage, it allows patients to stay on their medication a lot more efficiently. Next slide.
Biochips in business. So we have a couple of insights of the business of biochips. Biochips are expected to surpass $18.5 billion by 2024. And this is calculated from the entirety of the biochips usage found used throughout the world, from research to medical treatment to biochemical development and many other functions that it has in the health industry. Specifically, the microfluidics chip is expected to be the faster growing chip of them all in the market. The microfluidics chip may be the fastest growing chip as it is used as is the most applied between the three. It is mainly used to treat directly diseases found in genes, not necessarily for experimentation purposes or research purposes. So they will be the most commonly used found in general hospitals, the microfluidics chip. Next slide. Okay, and the future of biochips. Right now, they are researching into electronic DNA biochips used for powerful desktop devices, so they could simulate a biochip into a very powerful computer and run just purely through computational power, using very powerful computational power that is developed right now. And one of the biggest pushes for the biochip is to have a laboratory on a chip, which is like a very small experimentation smart card that can be used inside of an organism. It can analyze doctor surgeries, and then afterwards you can take it out and then put it into a laptop, and laptop acts as a terminal to interact with all of the experimentations done on that laboratory on a chip. And scientists are working to convert all their resources used to analyze organisms right on the chip. So it's they wanted to create the chips to be even more efficient and effective. And the lab on the chip would bring costs of resources down, so they could have tools more readily available for even more health professionals to use around the world. Next slide.
Okay.
So, yes, go ahead, Timmy.
Yeah, so this is where we just want to go ahead and thank you guys for watching the presentation. And if you have any, definitely feel free to ask in the Blackboard discussion board. And then, we do appreciate you guys watching the video and the time out of your day. And you have a great rest of your day.
Thank you.
All right, bye.